This is the lecture on prokaryotes, which is our week 2 topic in Bio 112. Okay, on the introduction page, what you're looking at is like an open, it's called a hydrothermal vent, but it's like hot springs. But it's like an opening in the earth where the water or heat rises up, and it's formed this little spring here, and there's a lot of growth of bacteria and archaea in this hot spring. And this is probably at Yellowstone National Park. I haven't checked, but I'm pretty sure that's where the it looks like, you know, those hot springs that are found at Yellowstone. Okay, so we have 3 domains of living things. The 3 domains of living things are bacteria, archaea, and eukarya. So 2 of the domains are prokaryotic. That means the cells do not have a nucleus. Okay, bacteria and archaea are prokaryotic. That means their cells do not have a nucleus, and they also are single-celled or unicellular. This is an image of a microbial mat, which may be the earliest form of life on Earth. Microbial mats form layers. They're like multi-layered sheets of prokaryotes, mostly bacteria, but some archaea. And they were probably found at hydrothermal vents, like deep in the ocean, or at the definitely in the ocean. Okay, so what you're looking at here are fossilized microbial mats called stromatolites, and you can see the layers really good in this in picture B. Little bit of information on the early Earth's early atmosphere. From what we know, from the evidence that we have, during the first 2 billion years of Earth's existence, the atmosphere was anoxic. You know, if you put A in front of a word, it means, you know, opposite. So what that means is there was no oxygen, no molecular oxygen, or O2, which is the form of oxygen that we breathe, that most living things need to live. So only organisms that can grow without oxygen, which we call anaerobic organisms, were able to live. Autotrophic organisms, meaning that they can make their own food, that convert solar energy into chemical energy, are called phototrophs. It means they carry out the process of photosynthesis to make their own food. And they appeared within 1 billion years of the formation of Earth, according to the evidence that we have. Now, what you're looking at is Cyanobacteria, which is kind of a greenish color, and you can see it along the edge of this little pond. Cyanobacteria, which is also known as blue-green algae, but it's not technically algae, because algae have eukaryotic cells. But the cyanobacteria evolved 1 billion years later, after the anaerobic phototrophs. And they began to add oxygen to the atmosphere. So that's what you need to know about cyanobacteria, is that they add a lot of oxygen to the atmosphere. They added oxygen to the atmosphere of the early Earth when there was no oxygen. Then we have extremophiles, and bacteria and archaea can be extremophiles. Either one can be, but a lot of the archaea are extremophiles. That means they live under extreme conditions. That can be really, really hot, really high levels of radiation, really cold, salty. In other words, in conditions that are not typically able to support life. some extremophiles actually thrive in the presence of acid, in acidic conditions that would normally kill other organisms. This is an image of, let's see if I can say it, Deinococcus radiodurans. And it has developed DNA repair mechanisms that allow it to reconstruct its chromosome, even if it has been broken into hundreds of pieces by radiation or heat. So it's an extremophile. It has adapted to high levels of radiation or heat. These are different types of extremophiles listed on the left, and the conditions that they grow in best are listed on the right. So acidophiles, pH 3 or below. Alkalophiles, because alkaline is basic, so that will be pH 9 or above. Thermophiles, phile, by the way, means like loving. So acidophiles love acid. Thermophiles love heat. So the temperature is hotter than what normally supports most organisms. You probably don't relate to 60 to 80 degrees Celsius as well, but in Fahrenheit it would be temperatures over 140, between 140 and 176 degrees Fahrenheit. And then you have hyperthermophiles, and that means that they grow best in even hotter temperatures than thermophiles. Psychrophiles grow at low cold temperatures. Halophiles are salt-loving, and osmophiles grow in high sugar concentrations. What you're seeing here, if you look at this is a colony of bacteria right here that's growing on this petri dish. Microbiologists typically grow prokaryotes in the laboratory using some kind of culture medium. In this petri dish, what you have is agarose gel that has some type of nutrient medium added to it. It's red, so it may contain blood. But anyway, you can also grow bacteria in a test tube in kind of a broth, like a liquid broth. So you can grow it on top of the solid in the petri dish, or you can grow it in a test tube. And we call that culturing. When we're able to grow bacteria or prokaryotes in general, we call that culturing prokaryotes. Can all prokaryotes be grown in a lab? Well, the answer is no. Most of them cannot. Over 99% of bacteria and archaea are unculturable. For the most part, this is due to a lack of knowledge as to what to feed these organisms and how to grow them. So, and some bacteria can't be cultured, if you look down here near the bottom, because they are obligate intracellular parasites and can't be grown outside a host cell. So organisms that are culturable can become unculturable under stressful conditions. And that is called VBNC state, viable but non-culturable. Okay, and biofilms are a little bit different than the microbial mats. It's still a microbial community, but it's not those layers like you see in the microbial mats. But a biofilm also has the bacteria that grow in this community secrete a gummy textured matrix that is primarily polysaccharides. And so that's a biofilm. Biofilms, by the way, they are good at protecting the individual prokaryotes that live in those communities. Okay, so these are the stages of biofilm development. I'll show you the pictures first. You can see it's pretty much this. You've got individual prokaryotes. These have flagella. And then you've got the small groups of these prokaryotes are secreting the matrix. And then they're just kind of building and building in steps 3, 4, and 5 until finally the biofilm becomes so large that some of the prokaryotes escape. So let's look at the stages again. There's the initial attachment to some substrate, some solid surface, and then there's irreversible attachment. So the initial attachment, they can be removed, or something can happen and they can become unattached. But once they reach step 2, the attachment to the surface is irreversible. They're attached to their surface by pili. These are like hair-like structures that extend out from their cell walls or from their capsules. And those pili anchor the bacteria to the surface that they're growing on. Stage 3 is maturation 1. That means the biofilm grows through cell division and recruitment of other bacteria. An extracellular matrix composed primarily of polysaccharides holds the biofilm together. So that's polysaccharides, remember, are complex carbohydrates. Then step 4 is maturation 2. The biofilm continues to grow and takes on a more complex shape. And step 5, the biofilm is starting to break down because, and some of the individual bacteria are escaping to colonize another surface. Sorry. One of the ways that we group prokaryotes is based on the shape of the prokaryote. The basic shapes are spherical, and that the word for the spherical shaped prokaryotes are cocci or coccus shaped. And then these that are kind of oval or rod shaped are bacilli or bacillus. And the ones that are spiral are sporili or sporillus. All right, this is just a typical prokaryotic cell. Prokaryotic cells don't have to have some of these structures, but they're always going to contain a nucleoid region that contains the chromosome. This is one chromosome, one big circular chromosome, but it's so long that it's folded all up in the center, in the nucleoid region of the cytoplasm. And then you have ribosomes that make proteins. Those are floating free in the cytoplasm. And then you have the plasma membrane. Outside that is the cell wall. And the cell wall of the of prokaryotes is composed of a material called peptidoglycan. We'll talk about that a little bit more. But then not all bacteria have this part, and that is a capsule with pili attached, and definitely not all bacteria have a flagellum. Mostly the ones that have a flagellum are the ones that move or swim. Okay, reproduction in prokaryotes is asexual. That means there's not a sperm and an egg where half of the chromosomes of one parent and half the chromosomes of the other parent come together and make a unique offspring. It's not sexual reproduction, it's asexual. And the process is called binary fission. Binary fission. When prokaryotes divide by binary fission, they're basically cloning themselves. But there are ways that prokaryotes can have genetic diversity. And those ways are transformation, transduction, and conjugation. And I see that our time's running out, so we'll start with transformation on the next section.